Good evening. We are Team DREAM, which stands for Generation Research in Using Alzheimer's Models. We are performing our research under the supervision of our mentor, Dr. Ricardo Araneda. We are combating the problem of Alzheimer's disease. This brain disorder not only leads to loss of cognitive functions such as thinking, remembering, and reasoning, but also gradually interferes with individuals' daily activities. It is the sixth leading cause of death in the United States and the third leading cause of death in the elderly. This lethal disorder affects the areas normally regulated by adult neurogenesis, which is the growth and development of new neurons in the olfactory bulb. Two major proteins involved in the Alzheimer's disease are the amyloid beta proteins and tau proteins, which we will go more into depth before our methodology section. The olfactory bulb is one of the first areas in the brain affected by Alzheimer's disease. One of the early signs of Alzheimer's disease affecting patients is the loss of scent, which is responsible in the olfactory bulb. This brain area will be the center of our study. In a Western society such as ours, there is a high rate of degenerative diseases and low levels of omega-3 fish oil in our diet. Omega-3 fish oils are found in fatty layers, in fish, also in plants, and nut oils and other fortified foods. It acts as an anti-inflammatory agent and can also help with learning and memory skills. And this is why, and the low levels present in our society, is why Team DREAM will be investigating how omega-3 levels in diets will affect the rate of neurogenesis.
So we have 3 research questions that we would like to answer for our project. Our first aim is whether or not omega-3 rich diet would have an effect on the rate of neurogenesis in the olfactory bulb particularly. And our second aim is whether the diet will have an effect on the cognitive activity and the behavior of the mice that we are researching. And then whether the increased levels will affect the development of Alzheimer's disease in the mice models.
So in order to answer these 3 aims, we'll be using a transgenic mouse model known as the 3XTG mouse, and this mouse has mutations in the genes that are responsible for the proteins that Melinda discussed, which are the amyloid beta protein and tau. And so amyloid beta is normally responsible for maintaining good synapse connections between neurons, and tau is responsible for stabilizing the microtubules of the neuron. So since both of these genes are mutated in the model that we will be using, we are expecting that these mice will have deficits in these functions within the neural system, and specifically for the functioning of the amyloid beta protein, the deficit usually occurs around 3 to 4 months. So that's the age at which we will be starting the experiments for the mice. As you can see in the table on the slide, we'll be having 4 groups, which will vary based on the type of mouse that they are, in addition to the type of diet they are receiving. So we will have 2 groups with the transgenic mouse, 2 with the wild type, and then 2 that are receiving the normal diet. And we will have an omega-3 supplemented diet, which will be provided through a type of oil that contains 2 types of omega-3 fatty acids known as DHA and EPA. In order to answer the question of the behavioral differences between these groups, we will be conducting a test known as the cookie test, which will be further discussed later, but basically its aim is to have the mice be able to distinguish between 2 odors that are pretty similar. And then in order to actually quantify the number of new neurons that are being generated in these mice, we will be using immunohistochemistry and antibodies that will allow us to actually observe the number of new neurons that are being integrated into the olfactory bulb. So this is our experimental timeline, and the times at the top refer to the age of the mice. So as I mentioned, we'll be starting when the mice are around 3 to 4 months old, because this is when the mutation has its strongest effect and begins to affect the mice. So we'll begin by doing 1 round of behavioral testing with the cookie test, and then after that is completed, the experimental groups in terms of diet will be started to be given the omega-3 supplemented food, and then about halfway through our experiments we will be injecting BRDU, which we will also go into more in-depthly later. but this is basically a way that we can track the new neurons that are being made. And the reason that we're doing it halfway through is because it takes about 30 days for new neurons that are being made to be fully integrated into the olfactory bulb. So we would have to do it at least 30 days prior to when we are sacrificing the mice in order to fully observe these new neurons that are being made. And then after 2 months of treatment, we will do a final round of behavioral testing, which will allow us to differentiate between the behavior in terms of olfactory learning in the mice before and after the treatments. And then finally, we will sacrifice the mice in order to collect the second part of our data, which is the quantification of the new neurons that are being made.
For behavioral testing, we are using the cookie test, which tests for association learning and olfactory memory and odor differentiation. First, the mice undergo 5 days of dieting, during which we take their weight daily and adjust the diet amount accordingly. Ideally, at the end of the 5-day period, the mice should retain 85% of their original weight. In preparation for the cookie test, 100 microliters of diluted L-carbone and D-carbone odors are placed on 2 pieces of filter paper. One of the odors is consistently associated with the cookie, which is the reward, by always placing the cookie underneath that filter paper with that odor. The odor locations are randomized every trial. Then the cookie test itself lasts 3 days with 5 trials each. On the last day, there is no cookie. This is because the purpose of the cookie test is so that we can ideally see the mice begin to differentiate between 2 different odors without the presence of the cookie, which is the reward.
So after the behavior test, we'll be quantifying the adult neurogenesis, and we'll be doing this using the technique called intraperitoneal injection. And that will consist of the BRU being injected into the peritoneum or the body cavity of the mice. So BRU is a synthetic nucleoside that incorporates into the cell during DNA replication, and that allows us to label the newly generated neurons. And we will be using IV injections because they have a fast absorption rate of the antigen BRU, allowing BRU to be more easily accessible to the proliferating cells.
The next procedure we will discuss is perfusion fixation. The goal of perfusion fixation is to rapidly and uniformly preserve the tissue in a lifelike state. And the brain is the center of our interest. The diagram in the upper right depicts the incisions that we made into the mouse after we administer anesthesia. The left ventricle of the heart will be injected with paraformaldehyde and PBS, which will assist us in stiffening the mouse and also draining of the blood, which will help to dissect the brain. And in the lower right of the slide, you can see a cryostat machine. This machine will be utilized to take brain slices. About 40 micrometers of the coronal section of olfactory bulb will be taken under a temperature setting of -60 degrees Celsius. Then we'll analyze these brain slices under a microscope.
So immunohistochemistry, let me break down the word for you. Immuno refers to the antibodies that we were using in the procedure, while histo means the tissue, which is the olfactory bulb that we were looking at. It is a procedure that we are using to stain the specific proteins in the tissue. It is an indirect method of immunohistochemical staining, and it is indirect because we are basically treating the cells with 2 layers of antibodies. The first being the primary antibodies, which will attach to the antigen S, which is BRU. And you can see in the picture, it's the antigen is the green triangle A, and the upside down Y is the antibody, the primary antibody. And then the second layer we'll be treating the cell with is the secondary antibodies, and these are the antibodies that have the fluorescent tags in them, which we will specifically see in the under the focal microscope. We are using the indirect method because it increases sensitivity to the protein binding, because there are 2 antibodies basically binding to the antigen, so we know for sure it's the targeted protein that we're looking at. Before we treat the cell with any of the antibodies, we are using a blocking serum to prevent any non-specific binding. And throughout the whole procedure, the cells are also rinsed with phosphate buffered saline, which rinses the cells so that when we see the image under the microscope, it's more clear.
After performing the immunohistochemistry, we'll be looking at our cells under the confocal microscope. So the BRD labeled cells will be labeling the neurons, which will appear red under the fluorescent light, and the NUN will be labeling all neurons, which will be labeled, which will appear green, as you can see in the picture. So the slices, so we'll be counting for the neurons that are marked both red and green, but then a rectangular area, and that will be looked under the fluorescent light. And several rectangular areas will be counted per layer of olfactory bulb. As of our current status, we are still training. We need to get through rigorous training in order to become proficient before we start with our mice, because our project is time dependent, and also our mice are very expensive. Right now, we are split into two groups. Two of us are performing the behavioral test and the IV injections, and the rest of us are doing the perfusion, immunohistochemistry, cryo-stat, and the confocal. And as for data collection, we're still in the process of collecting data.
So in regards to our team challenges, I think the biggest one has just been our small size and how it's been reduced, especially for this project because it's very lab heavy and we're working with animals, so it's also very time dependent. So we just have to work really hard at coordinating with each other's schedules and just making sure that, especially in terms of behavior, that we're able to go in because we have to maintain a very good timeline in order to perform the experiment properly. Additionally, the determination of our project was an interesting process, and just taking what all of us were interested in and aligning it with our mentor's research to come to a compromise is something that both we were interested in and our mentor was passionate about, and something that was feasible in terms of lab work and in terms of gemstone. And additionally, just maintaining a good system of communication within our team has also been important. And I think, in a way, it's been beneficial that we are a small team just because it's easier to communicate with each other and to get to know each other.
In terms of team dynamics, here are some advice to freshmen from Team Dream. First of all, propose and pick a project that you are personally passionate about. Team Dream's members are all extremely passionate about neuroscience, and this is why we picked this project, and we were sure that we wanted to do this project. Second, focus on the big picture of your project, as small details will definitely change in the process. Our project has definitely changed a lot from what it was freshman year to what it is now. Third, remember to compromise, practice courtesy, and make sure each member of your team has a voice. Fourth, remember that what you put into your research is what you get out of it.
So some future goals that we would like to, we have in mind right now are to finish our training quickly so that we can start our experiment. And we would like to start our behavioral test as soon as the mice are ready, so we can keep going and finish. And we will continue practicing the technique to prevent any mistakes in the future. And we would like to finish our thesis proposal and possibly publish our paper. So this concludes our presentation. We would like to thank our mentor, Dr. Ricardo and Aranita, our librarian, Jeremy Garantino, the Gemstone staff, and the audience. Thank you so much for listening. Questions?